Hello, I welcome you all to this series of discussions where we are left out with the properties or functions of proteins. So now let us discuss the functions of proteins. In the previous session, I had told you what are the basis of classification and what are the different categories of proteins. Proteins play a pivotal role in virtually all the biological activity and cellular constructions. Depending on their location in the cell and their physical and chemical nature, proteins perform a variety of functions. And let us sum up a few. The first one is they act like structural components. They are the structural components of cell membrane, cytoplasm, various organelles, extracellular materials and fibers. We have discussed protein keratin. That is a major component of our skin, hairs, feathers of birds, nails of different organisms, horns present in various animals. Similarly, the collagen protein is one of the important component of cartilages. In plants, the cell wall of the pollen grain is also made up of proteins, and that we will study in class 12. Many proteins act as the cytoskeletal structure like tubulin, and they provide rigidity to the cell. Second one is that proteins have catalytic properties and they act like enzymes. Obviously, we are going to study the structural proteins once again in this category. These proteins, they function as enzymes and they catalyze a wide range of biochemical reactions, and they play a very important role in the cellular metabolism. The next one will be that of the carrier proteins. So some proteins, they act as carriers and they can bind and transport specific ions and small molecules across the cell membrane. When I had taught you about active transport, facilitated diffusion, symport, antiport, then we have already discussed this. They control the flow of materials through the membrane. Hemoglobin, which is present in blood, or myoglobin, which is present in muscles, they transport oxygen from lungs to the peripheral tissue so that the nutrients can be oxidized. Lipoproteins, they carry lipids from liver to other organs. The next category will be that of the motor proteins. Under the carrier proteins, you will also have aquaporins, which are water channels present on plasma membrane. Motor proteins like kinesin, dynein. Remember the structure of centriole, cilia or flagella that I have taught you? There we have studied these proteins. They function in a variety of cell movements, including the locomotion by beating of flagella or cilia, intracellular transport of various organelles, separation of mitotic chromosomes by spindle formation, etc. Under this, you will also have contractile proteins like actin and myosin, which are comparatively more famous, I think. Once I talk about motor proteins, it is actin and myosin that comes to our mind fast. But kinesin and dyneins, they also help in cellular movement, but at the level of cytoplasm. So coming back to actin and myosin, they cause a variety of cell movements like contraction of muscle fibers, stress fibers, etc. Then next, they act as receptors. Some proteins function as the receptor molecules and they can bind and transport specific informational molecules like hormones, for example phytohormones like auxin, and they regulate their activities. Hormones like insulin, the thyroid hormone, the growth hormone are also proteins which regulate specific metabolic activities, and that's why they are called as the regulatory proteins. Next, let us come to the more famous category that is otherwise called as the antibodies. During this COVID time, antibodies need no further explanation. They are otherwise called as immunoglobulins, and they participate in the defense mechanism and they provide immunity to the body. This is the fact which you have been studying since your lower classes. Antibody proteins have requisite specificity. Why they are able to recognize the specific antigens? Because they are quite specific at it and they can distinguish one, you can say one protein or antibody can distinguish its own specific antigen from others. They recognize the invading foreign particles like bacteria, viruses, Under this, you will be having fibrinogen, thrombin, which are the blood clotting proteins also. And they prevent excessive loss of blood by forming the clot when our system is injured. Snake venom, bacterial toxins, toxic plant proteins, they are also proteins among the defensive functions. So when proteins, we say that they are defense proteins, don't let us concentrate our discussion only to the antibodies. You should also include the clotting proteins, the snake venom proteins, bacterial toxins or plant toxins, because they are also defensive in nature for the respective organism in which system they are present. Some proteins we have discussed, they act like storage proteins. In that case, I had told you glutenin, that is present in wheat grains, ovalbumin, that is present in the egg white, and casein, which is present in storage proteins. Then proteins also act as sensors. They can regulate the flow of substances. They can act as sensors or switches in response to stimuli and communication of information. In nervous system, channel proteins are present. They sense multiple stimuli and act as a logic gate. Proteins contain multiple sites that communicate with each other. A conformational change is induced by binding of the molecule to one site in a protein, and that can change the other sites. Remember nodes of Ranvier that we have discussed about, or the surface proteins that are present in the synaptic cleft. They are the examples under this category. Under the last, you can say, function, we can study that proteins can change energy from one form to another. For example, myosin. It is a protein which is present in our cytoskeletal structure. It can bind to the active filaments at one end, and it also has having a snake-like structure. Remember I told you, and it is having on its head two receptor sites, and one of them is for binding to the ATP molecule. So it can convert the energy in the form of ATP to mechanical energy so that the actin and myosin filaments can slide over each other. So having said this, we have already come to the end of the discussion on proteins. Now let us move over to the next important group of biomolecules, that is the lipids. Lipids are fatty acid esters of alcohols or the related substances, and they are generally insoluble in water, but they are quite soluble in a number of non-polar organic solvents like ether, benzene, chloroform, acetone, etc. However, they can form colloidal complexes and they can get dispersed uniformly in water in the form of minute droplets, and once they are dispersed in the form of minute droplets, the complex is called as emulsion. So lipids, one of their important characteristic that they can form emulsion. That is why the fat that you are eating in your diet, that is digested by your body. Unless you emulsify it, you cannot do it. And in lower classes, you have studied that it is your bile juice, and particularly the bile salts that are present in it that help in emulsification of fats. Lipids are basically made up of carbon, hydrogen, nitrogen, and sometimes oxygen. And oxygen is always very small in its amount if you compare its relative proportion to that of hydrogen and carbon. And many lipids contain small amounts of additional elements. Sometimes you will find phosphorus, sulfur, etc. Nitrogen, if you compare, then in proportion to proteins, these fats have less amount of nitrogen. Lipids, they consist of a structure where you will be having a head and there will be 1, 2 or 3 such zigzag tail-like structure. So this part is polar and this tail part is non-polar. So if I represent a fat molecule or a lipid molecule, then the diagrammatic representation will be like this. The polar or the hydrophobic part which is attracted to water is called as the head. And the 3 non-polar zigzag chain-like structures, they are the hydrophobic portions which are repelled by water. Already when we were discussing amino acids, I had told you that if a particular molecular structure has a hydrophilic and hydrophobic moieties present to it, then its nature is said to be amphoteric or amphipathic. So these fats are amphoteric or amphipathic in their nature. Now let us come to this zigzag tail-like structure, which are otherwise called as the fatty acids, and they are one of the basic components of fats. So once we are talking about the fatty acids, they are the organic acids which have hydrocarbon chains that end in carboxylic acid groups. So the hydrocarbon chains, they may be straight or they can have a ring structure. And some fatty acids can also have OH or hydroxyl groups present in them. So basically they will be having a CH3, CH2, CH2, CH2. This intermediate CH2 structure will be variable, and at the last they will be having a COOH. Sometimes double bonds can be present in the straight chain and sometimes double bonds won't be present. Depending on that, fatty acids can be classified into two main types. The first one is said to be saturated. Already these terms you are quite familiar with, because nowadays we are becoming very health conscious and we know that this saturated fatty acids that are not good for our health, whereas non-saturated ones are more conducive for your health. So saturated fatty acids don't have in any double bonds in their carbon chain. So whatever I have written over here, this is a saturated fatty acid, and they have the general formula which is said as CnH2n and O2. CnH2n and O2. For example, you will be finding palmitic acid that is given in your NCERT book. So it is according to this C16H32O2. Sorry, H32O2. This is palmitic acid. And you have another example also that is called as stearic acid, that is obviously C18H36O2. You remember this general formula, it will be easy for you to remember. This is the molecular formula for the fatty acid stearic acid. Now let us come over to unsaturated fatty acid. Definitely unsaturated fatty acids will be having more or one double bonds in their carbon chains, and the general formula is like this: CnH2n minus 2xO2. This is for unsaturated fatty acid, and they will be having double bonds in their structure. Unsaturated fatty acids have lower melting point than the saturated fatty acids. That is why now it is almost the time of winters. How you can by looking and differentiate which one is saturated fatty acid and which one is non-saturated fatty acid containing lipids. The lipids which solidify at room temperature, like that of coconut oil, butter, margarine, those cases you will be finding that they will be having saturated fatty acids. That is why they easily tend to settle down. The ones which remain as oils or in the liquid state, even if the outside temperature is less, they are generally unsaturated fatty acids containing fats. Unsaturated fatty acids, they have a lower melting point than the saturated fatty acids. Most plant lipids have unsaturated fatty acids, and most animal lipids, they have saturated fatty acids. Having said this, now let us move over to the classification of lipids, how lipids are classified. So lipids are mainly classified like we have classified the proteins. It is simple lipid. As it was simple protein, in protein we were using the word conjugated. Here we will write the word complex lipid, and the third category will obviously be the derived lipid. Okay, so the three categories of lipids will be simple, complex and derived. Simple lipids are the esters of fatty acids with alcohol, and they are of two types. One is neutral fat and second one is wax. Neutral fats are otherwise called as the true fats. The neutral fats, they are neutral because they don't have any charge. I think that is quite obvious to you, and they are composed of carbon, hydrogen and oxygen, but they have fewer oxygen atoms in comparison to carbon atoms. They are also called as triacylglycerols. A fat molecule consists of two components, that is one molecule of alcohol and three molecules of the same or the different long chain fatty acids. A glycerol has three carbons and each one is having a hydroxyl or OH group. Though we have discussed this previously, still let me remind you this one more time. I think glycerol formula all of you remember. This is CH2OH. Already in one of these videos I have said this. So either this palmitic acid or stearic acid, instead of writing this long form, I can write it the carboxylic, like I will write it as CH3, then CH2 whole something, and then COH. Instead of writing as this CH3 CH2 part, I am writing it as R, and this COH I am writing in the reverse side. So this is COOH R1. This is my first fatty acid. This COOH R2, this is the second fatty acid. COOH R3, this is the third fatty acid. So I will be taking, actually I should have written it in the other orientation. Let me write it once again. In this way I cannot show the condensation. Let me write the COOH group as HOOC R1, HOOC R2 and HOOC R3. Now you condense this and this. You will be getting 3 molecules of water out of it. And the resultant structure will be CH2O, C double bond O, R1, then CHO, C double bond O, R2, then CH2O, C double bond O, R3. So this is your triacylglycerol. I hope you remember. Already I have taught you this. The second one, now let us come to the category of waxes. Waxes are the lipids which are generally composed of long chain saturated fatty acids and a long chain saturated alcohol that is of higher molecular weight, but the alcohol will not be glycerol, as simple as that, as I have told you right now. The example under this category will be bee wax. Bee wax is the secretion of the abdominal gland of worker honeybees, and it is a thick and tenacious material which is almost behaving like plastic at room temperature, if you might have marked it. The second one is the sebum. Sebum is your own body's secretion, the oily secretion which keeps your skin soft and supple. It is a fatty secretion of the sebaceous glands of your skin. Cerumen, that I have told you already when I was teaching you the external structure of ear. Our ear has various ear glands or ceruminous glands which are lining our external auditory canal. They produce a brownish, soft, waxy secretion which traps dust and dirt that tries to enter with the air into your auditory meatus. And the fourth one is paraffin wax. And this is actually not organic in origin. In petroleum industries, when you fractionally distill petroleum, or petroleum undergoes fractional distillation, then your candles are made up of it. So they are not natural wax, but they are the fractional distillation product of petroleum. Then now let us come to the complex lipids. So complex lipids are the derivatives of simple lipids, and they have additional groups like phosphate, the nitrogenous base, the protein, etc. And they are derived further into the first category, which is called as the phospholipid. The name itself indicates that it is having a phosphate group attached to a molecule of glycerol or other alcohol, and it is joined to one of its outer hydroxyl groups, and two fatty acid molecules are linked to the other two OH groups, and a nitrogen containing base is bound to the phosphate group. The phospholipid molecule has a strongly nonpolar or hydrophobic tail that consists of fatty acid chains and a polar or hydrophobic head, which comprises of negatively charged phosphate group and positively charged base. Again, phospholipids are of two types. One is called as glycerophospholipid and another is sphingophospholipid. So here you will be finding that glycerophospholipids, or they are also called as phosphoglycerides, that contain glycerol as their alcohol part. So here if I try to draw this, then there will be, the structure will just be like this. If this is the choline group, then it will be attached to a phosphate group, then it will be attached to the glycerol molecule, and here will be the two chains of fatty acids. So if you simply remember the structure of phospholipid, then it is a structure of this. So having said this, let us come to the example, that is this glycerophospholipids, lecithin, cephalin, they will be the example under this category. Then second one is sphingophospholipids. They are present, or the name itself indicates that their alcohol part is called as sphingosine. That is why they are called as sphingophospholipids. The second category of complex lipids is the glycolipids. The name itself indicates that they will be having a carbohydrate group attached somewhere over them. The glycolipids, they contain ceramide, and generally that is attached to the fatty acid or one or more sugars, particularly galactose, and they are generally the components of the cell membranes, particularly the myelin sheath of nerve fibers and the outer surface of the nerve fibers, and if I am remembering it correctly, then they are also present in the membrane of chloroplast. Cerebrosides are the most simple examples of glycolipids. So glycolipids composite, or they are the composite structures of myelin sheath. Then third one is lipoproteins. Obviously they are proteins associated with lipids. So glycolipid example is myelin sheath. So lipoproteins, they contain the lipids and proteins in their molecules. They are the main constituent of the cell membranes. Lipoproteins are generally present in, you can say, egg yolk or milk. Lipids are transported in the blood plasma, and lymph has lipoproteins. They are the transport vehicles for lipid circulation. And here comes the most important part of this, and that is otherwise called as lipoproteins are classified into the following categories also. They are also called as chylomicrons. This you will study in your digestive system. Once fats are digested, they are transported into the lymph as chylomicrons when they have to cross the intestinal wall. Second one, this name you have studied when you might have got for a blood test, or when you have got a blood test done, you might have heard VLDL term. They are called as very low density lipoproteins. They are the second category. The third one is low density lipoproteins, LDL. The fourth category is HDL, high density lipoproteins. And the fifth one is free fatty acid albumin complex. So they are the 5 categories of lipoproteins that are present in your body. Then let us come to the next category, that is that of the derived lipids. And this is the most diverse group among the lipids. The derived lipids are the derivatives which are obtained on the hydrolysis of either simple lipids or the complex lipids. The most famous example will be the steroids. This term is quite popular among you. The next one is terpenes. And the other one that is quite common is prostaglandin. Once you study the human reproductive system, prostaglandin will be more clear to you. So generally the derived lipids, or let us come to the first category, that is that of steroids. Steroids are compounds which contain a cyclic steroid nucleus, and the name of that ring is a little bit tongue twister. It is called as cyclopentanoperhydrophenanthrin, or in short it is called as CPP ring. Let me write it down. I am writing it separately so that you remember it. From here we take C, then pentano. From here we take the first P, then the third one is per hydro. From here we take another P, and the last P comes from phenanthrin. So it is CPPP or CPQ in short. So now let us write down the structure of it. It might seem complicated to you, but it is not that complicated. Sorry. If I just write the skeletal structure, it will be having 2 hexagonal rings attached to it. Here there will be some side chain substitution. Then another hexagonal ring which will be attached to this area, and the last one will be a pentagonal ring. This is 1 2 3 4, and here also there will be a side chain substitution. But let me write it down a little bit more in details so that you don't get confused, right? So for cholesterol, let me write the structure. Though the structure appears a little bit more complicated, but with a little practice you will be able to know it or quite master it. The steroids do not contain any fatty acids, but they are included in the lipids because they have fat-like properties. They are not straight chain compounds, as you can see. Rather, they are composed of 4 fused carbon rings. There are several steroids that you can find in a biological system. They include cholesterol that I have drawn over here. Then there are also other examples here: bile acids, vitamin D, the sex hormones, the adrenocortex hormones, the cytosterols, the cardiac glycosides, alkaloids, they are all examples of the steroids. Having said this, let us discuss the cholesterol molecule in a little bit more detail. So here the cholesterol is the most abundant steroid in the animal tissues, and it is present in the food, especially that is rich in animal fats, and it is also formed in liver itself, and the formula is C27H45 and OH. This is cholesterol, and it also, cholesterol along with its esters are insoluble in water. That is why they are deposited on the lining of the blood vessels and we get hypertension and other problems. Let us come to the second category of derived lipids. They are otherwise called as the terpenes. They are lipid-like hydrocarbons which are formed of isoprene rings. So isoprene rings are C5H8 rings, or units rather. Terpenes are major component of the essential oils which are produced by certain plants. These essential oils, they are generally with two or four isoprene units. Camphor, then menthol of pudina leaves, gibberellins, they are a group of plant growth hormones with four isoprene units. An equal number of isoprene units are also present in vitamin A, vitamin E, vitamin K. The phytol or the tail portion of chlorophyll A molecule is also a terpene, and it is having four isoprene units. The carotenoids, which are present in tomatoes, carrots, and give them the particular colorations, or the yellow color or orange color of marigold, is due to xanthophylls. So these carotenoids and xanthophylls are all terpenes, and they have 8 isoprene units. The neutral and synthetic rubber are also terpenes with thousands of isoprene units which are arranged in linear fashion. Now let us come to the next category, that is the prostaglandins. So prostaglandins are the hormone-like compounds which are derived from the 20 carbon polyunsaturated fatty acids such as arachidonic acid, which is given in your NCERT book. And their richest source is the secretion of the seminal vesicles in males, and during our menstrual cycle the fluid which is secreted, that also contains sufficient amount of prostaglandins. They have a variety of biological effects. They can dilate your blood vessels and cause vasodilation. If required, then they can show the antagonistic activity, that is, they can constrict the blood vessels, and they can also say or they can also be acting like vasoconstrictors. They can stimulate the smooth muscles of the intestine so that food can move properly. Perhaps they act by regulating the production of cyclic AMP by adenylate cyclase, that we have studied during the mechanism of hormone action. The last part is your lipid peroxidation and antioxidants. These are the terms which are nowadays very popular among us. Peroxidation means the auto-oxidation of lipids when they are exposed to oxygen, and it is responsible not only for the deterioration of the quality of foods. The foods become rancid, so that is due to this lipid peroxidation. It also damages to the tissues if it is occurring inside your system, where it can be a cause of cancer or inflammatory diseases or atherosclerosis. It also brings aging, means you will age faster by this process. The negative effects are considered to be caused by the free radicals like ROO minus, RO minus, HO plus, etc., which are produced during the peroxide formation from fatty acids. Generally, the fatty acids which contain methylene interrupted double bonds, that is, which are found in the naturally occurring polyunsaturated fatty acids, they show this mechanism more. Lipid peroxidation is a chain reaction, and it provides a continuous supply of free radicals that initiate further peroxidation, and that is why the effect is so devastating and chain-like. We can also control and reduce lipid peroxidation. It is not such a gloom scenario. In humans, their activities and nature, they can be controlled by the use of antioxidants. And the antioxidants which are very popular are propyl gallate, butylated hydroxyanisole, which is otherwise called as BHA, then BHT, which is butylated hydroxytoluene, They are famous antioxidants which are used as food additives. I think these terms might be sounding a little bit more complicated. Ever you have seen that when you buy a chips packet, inside the chips packet there are certain things which are mixed to those potato chips, which are deep fried in saturated fats so that they remain fresh for a longer period of time. If you see the backside of the wrapper, you will find the name of these antioxidants that I am telling about. So when you are frying your potato chips in fats, that means you are making them prone towards lipid peroxidation. But if you want to make them fresh, or if you want to use them fresh after a long period of time, then you should add these names like BHA or BHT, which I have said. Then they will remain fresh. I think now it is clear to you. Naturally occurring antioxidants, generally they are the vitamin E. So that is why it is said that if you apply the face creams which contain a lot of vitamin A, that makes your skin stay longer for a longer period of time. It is because it prevents the lipid peroxidation. Vitamin E is otherwise called as tocopherol, and that is lipid soluble. Another compound is urate, as well as vitamin C. Nowadays you have seen models and others, they are advocating that you should apply vitamin C directly onto your skin. It is because it is a strong antioxidant. But vitamin C is water soluble, and beta carotene is also an you can say antioxidant, but it is at low concentration of carbon dioxide. If you oxygen, if you increase the concentration of oxygen, then beta carotene, it will get oxidized and it will no longer act as antioxidant. So having said this, we have come to the end of our discussion on lipids as the biomolecules. Thank you.